Well, of all the crops in California, almonds probably receive the most attention. And as I talk with people, the two things they want to know about is nitrogen and salinity. So we're really lucky today to have Daniela Reineke with us. Daniela is a PhD student at UC Davis in the Soils and Biogeochemistry department. She's working with salt and nutrient transport in soil and modeling those processes. And so she's looking at irrigation and fertigation management and how those can be manipulated to manage salt and nitrate leaching. Dr. Patrick Brown is a regular speaker at our FREP conference, and Daniela is working closely with Dr. Brown, and so it's a sort of a team report, and we get to hear from Daniela and get to know her better this time. So Daniela, we're glad to have you with us and speaking about improving nitrate and salinity management strategies for almonds.
Good morning, everybody. So today I'm going to talk about two different things. So the first is a field experiment that we are conducting on improving nitrate and salinity management strategies for almond. And the second thing is greenhouse work that a former postdoc, Francisco Valenzuela, has done. And the results from these experiments is actually what led up to this field experiment later. So this slide shows a map of soil salinity in the western San Joaquin Valley. And on the map, everything that's in blue means a soil salinity of 0 to 2 decisiemens per meter. And all other colors mean values higher than that. So we can see that soil salinities greater than 2 decisiemens per meter are fairly common in the western San Joaquin Valley. And although this value doesn't seem to be very high, commonly cited value of the salinity threshold of almond is only 1.5 decisiemens per meter. So almonds are pretty salt sensitive, which is why in some regions of California we have soil salinity causes a problem to almond production. The way we usually manage salinity is by applying an extra amount of water called the leaching fraction to move salts, in this case represented by chloride, down and ideally out of the root zone. And the more water we apply in excess of the crop's demand, the lower will be the salinity in the root zone for a given irrigation water salinity, which can be seen in this graph on the right-hand side, which shows soil salinity over soil depth for different leaching fractions, where this bottom graph here indicates the highest leaching fraction, and the top graph here indicates the lowest leaching fraction. The problem is when we leach salts, we may also leach other ions, such as nitrate, and that's because the transport behavior of nitrate and chloride in the soil is fairly similar. So both ions are moved with the water as dissolved ions, and both are anions that don't interact much with the matrix, so they are fairly mobile in soil. So whenever we do something to leach salts, we are always at a risk to leach nitrate as well, which may lead to environmental problems, for example, if nitrate contaminates the groundwater, and also to economic losses because we reduce the nutrient efficiency. So whenever we think about strategies to manage salinity, we always need to keep this trade-off between salinity management and nitrate management in mind. There has been quite a bit of research on salinity management, but most existing guidelines are really based on conventional irrigation systems, such as flood irrigation. And as you can see from these two pictures on the right-hand side, flood irrigation is much different from drip irrigation, which is mostly used today. So for flood irrigation, we distribute the water uniformly across the whole soil surface and try to keep the keep salinity low until a certain depth. And for drip irrigation, we really only wet a small fraction of the soil surface. So there's a very concentrated zone of leaching in the center that is free of salts. But then in the margins to the left and to the right, soil salinity is really high. So a plant that grows under these conditions experiences a high degree of heterogeneity in soil salinity. And the good thing is that we can actually control a little bit what these spatial distributions look like by the way we irrigate. So with our project, we want to answer the question, how can we reduce salt damage while simultaneously preventing nitrate leaching under these heterogeneous conditions? So yeah, first we can see that all options got some votes, which indicates that the salinity problem really varies by location. But most people apparently thought that the problem was at least moderate. So most people seem to have experienced the problem in some way. So the way we approached these questions had two parts. The first part really focused on plant physiology. So how do plants respond to heterogeneous salinity conditions in the root zone? And this question was addressed by using split root experiments by a former postdoc, Francisco Valenzuela. And in the second part, we wanted to know how can this information be used to improve salinity and nitrate management using drip irrigation. And in this case, we chose a lysimeter approach, which has some characteristics of a greenhouse experiment because we grow our plants in closed containers, which gives us some control of what's happening in the root zone. But also the experiment is conducted outdoors, so the plants can actually grow under outdoor conditions and grow to full size. So in order for Francisco to study the plant response to heterogeneous root zone conditions, he had to find a way to translate these very complex patterns of heterogeneity to an experimental design that was simple enough that he could still measure in detail how much water and nutrient is taken up at which location at which time. And the way he did it is by using a split root experiment. So he split the root system into two parts, side A and side B, and each part was put into one separate pot with a nutrient solution. And this way he could expose the different parts of the root system to different concentrations of nitrate and also salinity. And then this approach also allowed him to measure water uptake from each site, which then served as an indicator for him to determine root activity under the different conditions. This slide shows the different treatments that were imposed in the first two experiments. So in experiment 1, there was only salinity heterogeneity imposed. So site A contained a nutrient solution, and site B also contained a nutrient solution, but also sodium chloride was added to the solution. Experiment 2 was on nutrient heterogeneity, where no salinity stress was imposed. So site A contained a nutrient solution and site B was nutrient depleted. So either all nutrients were lacking, it was just the eye water, or only a specific nutrient was missing. And here we see the results of the effect of salinity heterogeneity. So what this graph shows is the proportion of water taken up from each site, so over time. So in the beginning, both sites take up 50% of the water, but as soon as the treatment starts at time 0, we immediately see in effect, so the uptake from the nutrient and salt site is reduced, and we have preferential uptake from the nutrient site. So the ratio is about 60 to 40%. And then Francisco also wanted to know, is this response reversible? And if so, does it take a long time for uptake patterns to change when conditions change? For example, does the plant first have to grow new roots that would allow it to take take up more water from one side than the other? Or would it be kind of an immediate effect? And in order to test that, he did a variation of this experiment that started exactly like the first one. But after day 3, the sites were switched. So the root system that was first that was initially in the saline site was switched to the non-saline site and vice versa. And when we look at the results, we see that the response is very quickly. So the root uptake pattern changes immediately to the nutrient site again. And this shows that the response is quickly reversible when conditions change. These are the results for the nutrient heterogeneity. So we clearly see a preferential uptake from the nutrient over just the eye water, which makes a lot of sense because the plant needs to be able to acquire the nutrients from there. And then Francisco also wanted to know whether which ion is actually causes this response. So whether it's maybe all ions or whether it's only specific ions that cause this effect. And here are the results for the ones that he found to be most important. So potassium and nitrate. So in this case, the nutrient depleted site contains all nutrients except this one nutrient. And what we can see in both cases, there was preferential uptake from the nutrient site. And this effect is a little bit bigger for potassium than for nitrate. So what we've seen from the previous experiments is that we have preferential uptake from the site that is not saline and from the site that is not nutrient depleted. But the question now is what happens if the site that is non-saline is also depleted in nutrients. So and the application in the field is what happens if the non-saline zone gets depleted in nutrients and all the nutrients are maybe leached into the lower saline zone. And in order to answer this question, Francisco added a third experiment where now there was a saline site that also contains nutrients, and there is a non-saline site, but this site is depleted in nutrients.
All right, Daniela, what was the happier root system in this scenario here?
I'll answer that with the next slide, but yeah, first of all, I want to point out that preferential uptake from the two sites almost got the same number of votes, which I think makes sense because intuitively, I think both results would be reasonable. So I'll show the actual result in the next slide. So these are the results. So what we can actually see is that we now have preferential uptake from the saline site. So from the nutrient side, despite the fact that there's also salinity there. And so this means that the presence of nutrients in the saline zone and absence in the non-saline zone increases root activity in the saline zone. And Francisco also, again, did this specifically for potassium and for nitrate. And here for potassium, we see a very similar effect. So there was preferential uptake from the saline site starting at day 3. For nitrogen, the response wasn't that clear. So initially there was preferential uptake from the nutrient depleted site. But at the end of the experiment, the uptake from both sites was almost equal. So it might indicate that had this experiment been run longer, there might have been a switch at some point. So what does this mean now for salinity and nitrate management? So first, water uptake distribution is controlled by both the nitrate and salinity distribution. And so leaching of nutrients into the saline zone may lead to problems for 2 different reasons. So first of all, the nutrients in this zone may just not be available to the plant because it doesn't use this zone. And second, if nutrients become depleted in the root zone, the plant may be encouraged to explore the saline zone. And that's problematic because if water uptake is increased from the saline zone, then the plant is also exposed to the toxic ions, so sodium and chloride, that will taken up more and may cause salt damage. So to illustrate this a little bit more, I have here three different possible distributions of nitrate in a saline root zone. So for all of these scenarios, we assume that salt is kind of pushed out towards the margins, but by the irrigation water, which is kind of seen by the salinity distribution indicated by the heat map in the background. And then the nitrate, which is indicated by the red dots, can either be uniformly distributed in the root zone. It can also be mostly in the low salinity zone. It can also be only in the saline zone. And if we wanted to rate these scenarios according to which one is most beneficial, we would probably say that the absolute worst case is having the nitrate only in the saline zone, because in this case, the plant, in order to take up these nutrients, the plant would need to use the saline zone and may take up a lot of sodium and chloride. And the best case would be scenario B, where nutrients are kept in the low salinity zone. So this way, the plant can preferentially use this zone and maybe is not so much affected by the higher salinities below. And so the question is, how do we achieve these conditions as in scenario B? And our hypothesis is that we can best achieve this if we have a high irrigation and fertigation frequency. So the high irrigation frequency would cause this non-saline zone to always be moist, so that the plant would have water available. And the high fertigation frequency would make it possible that we apply nitrate in very small amounts per day only, so that the plant is actually able to keep up with taking up the nutrients before they have time to get leached out into the saline zone. In order to test this experiment, we have started a field experiment which tests the hypothesis whether a high frequency irrigation and fertigation approach would reduce salt damage and nitrate leaching. And we contrast two different scenarios here. So first is a low frequency scenario with irrigation every 4 days and fertigation every 8 days. And the next one is a high frequency scenario where we irrigate and fertigate every day. And then, so these scenarios will be irrigated with saline water, 1.5 decisiemens per meter. And then we also have a non-saline control for the high irrigation frequency treatment. This experiment has 3 different components. In the first step, we want to only characterize the spatial patterns of nitrate and salinity that develop in the soil. So we will take pore water samples and soil samples to determine that. In a second step, we want to look at the plant response to these treatments. So we will look at plant growth and yield. And in a third step, we want to use a numerical model and see if we can actually predict those patterns that we observe in the lysimeter experiment. So the experimental design is a factorial design, and we have 3 different irrigation and fertigation treatments, 2 different soil types, and 4 replicates, which results in 24 lysimeters. And they are arranged the way as in this drawing on the left-hand side. And this is a cross-section through a lysimeter. So we have 16 pore water samplers that we use to extract soil solution and then analyze it for nitrate concentration and salinity. And they are arranged in a grid-like pattern. And then we have 3 nutrient probe access tubes for measuring water contents at different depths. And then each lysimeter also has a drainage system that allows us to collect all the drainage outside of the lysimeter and analyze it for quantity, salt concentration, and nitrate concentration. And on the right-hand side, you see a picture of the lysimeters. So there are 2 trees with 2 different rootstocks in each lysimeter. and the other picture shows the soil surface with a different instrumentation. So this research is still ongoing, so we don't have final results, but what we've seen so far is that soil solution salinity has increased to an average of about 6 to 8 decisiemens per meter in the saline treatments. And as is expected, salinity is close to the irrigation water EC of about 1.7 decisiemens per meter under the emitter, but reaches salinity of about 10 decisiemens per meter farther away from the emitter. And so far, we have not observed any significant differences in plant growth in the different treatments. With the modeling, we are also still at the beginning, but what we want to do is actually translate this reality that we observe in the field into a numerical model. So we are actually simulating this block of soil, and we are simulating water flow, nitrate transport, and salt transport. And so this picture actually is output for a simulation of soil moisture. And what we want to achieve with this, so this is actually an existing model, but this model doesn't account for all of these processes that Francisco observed in his split root experiment. So the process of root uptake is very simplistic in this model. And we believe that in order to make accurate predictions, we need to account for all these processes that we know of. And on the right-hand side, we see the results of simulating electrical conductivity. And even though this is still the original model and it hasn't been calibrated at all, we can already see that we can see some characteristics that we also observe in the field. So in the vicinity of the drip emitter, salinity can be pretty low, like about 1 to 1.2 decisiemens per meter. But then in the margins around those wetted volumes, we see that the salts, the salinity really gets high up to over 10 decisiemens per meter. So in summary, plants' uptake patterns are controlled by both salt and nutrient distribution. And microirrigation results in highly non-uniform salinity patterns. And developing guidelines that take heterogeneity into account may help to reduce salt damage and minimize nitrate leaching at the same time. And I want to thank all people who have helped with this project, and thank you very much for your attention.
Thank you, Daniela, for that interesting presentation. Really some great research you're doing.